for joining, everybody. My name is Jeremy Warner. I'm a steering committee member of the COVID-19 and Cancer Consortium, and this is our first scientific retreat. I'm going to go through a few slides in the beginning, and then I will turn it over to our keynote speaker. For those of you who might not be familiar, this is our mission statement, which is that the driving goal of the COVID-19 and Cancer Consortium is to collect and disseminate prospective, granular, uniformly organized information on people with cancer who are diagnosed with COVID-19 at scale and as rapidly as possible. We were founded in March of 2020, so just about a year ago, and we have grown rapidly since then. We now have 128 institutions that are participating in the consortium, and a whole lot of people. So this is, as of yesterday, every active participant, and this is just about 500 names. And so thank you all. If you see yourself on this slide, thank you for your involvement. We also have two really important teams that make our consortium function. We have a project coordination team at Vanderbilt University Medical Center, where I'm also located. And that is headed by Sanjay Mishra, who's our project coordinator since the beginning. We have biostatisticians, logistics, database analysis, and clinical research informatics expertise. And then we have a steering committee that's composed of 11 individuals that you can see on the right. Our main product is the registry. So we collect cases of patients with cancer and COVID-19. We began again just about a year ago. After a brief period of very few reports coming in, we've really grown quite quickly to the point where we're now well over 9,000 case reports and continuing to see them come in as a pretty good clip ever since the beginning. We've also been productive, so we've published a number of papers. And this is a fairly complete list, including our most recent publication, which came out last week in the Annals of Oncology. And that was led by Petros Grievous. And speaking of which, now we're going to move on to the content of the session. And Petros is going to introduce our keynote speaker. Petros, are you there?
Hi, Jeremy. Hi, everybody. Can you hear me well?
We can.
Wonderful, wonderful. Thank you, Jeremy, for the introduction, and thank you for being the machine and the huge collaborator in this. My name is Petros Grievous. I'm an associate professor at University of Washington and Fred Hutch and SCCA, Seattle Cancer Care Alliance. And today I'm really honored to be part of this amazing initiative. Thanks to everybody for being part of this. This is a huge volunteer effort that now it's 1 year old, and as Jeremy mentioned, we're all proud of the work being done, and all the work is dedicated to the patients and families affected by this pandemic and COVID-19 disease. I would like to have the honor and really excitement to introduce the keynote speaker for today, Dr. Stephen Pergam. Dr. Pergam is my close colleague here at Fred Hutch and University of Washington Seattle Cancer Care Alliance. He received his medical degree from the University of Nebraska, his infectious diseases training at the University of Washington, and his master's in public health, also from the University of Washington in the public health in school. He's currently an associate member in the clinical research and vaccine and infectious disease divisions at the Fred Hutchinson Cancer Research Center. He's an associate professor in the Division of Allergy and Infectious Diseases, and also an adjunct associate professor in the Department of Epidemiology at the University of Washington School of Public Health. Among multiple other important duties and endless work, Dr. Pergam is part of the FDA committee that votes on the COVID-19 vaccines, and he's very interested in vaccines in immunocompromised patients. And I can tell you, his research is well recognized internationally with huge impact in the field, in the field of hematology oncology, and in the past year in the field of COVID-19. I just would add one personal word. Dr. Pergam has been such a compassionate colleague with empathy for patients, and a tremendous resource for all of us. And I still remember the beginning of the pandemic, himself and other colleagues here were like the lights that helped us go through this. and help us establish guidelines. Without further ado, thanks everybody for joining. Thanks everybody for tremendous volunteer work in the past year. We keep going here to help patients. And Steve, the floor is yours. Thank you for joining us. Thanks for all your work.
Thank you so much, Petros. That was a really wonderful introduction. Hopefully you guys can see my slides. Just want to be sure.
Yes.
Okay, great. So first of all, at the beginning, I'd like to say thank you to the committee for asking me to be here. It's a real honor to be presenting to a group who I think has done some of the most seminal work in this field. The way that you guys have accomplished the CCC19 process and collected data is really enviable. And I think something that's been really important for many of us who've been trying to advocate for our patients for different reasons. So I really excited to hear about the next stages and where you go and to participate in this conference beyond just my introduction here at the beginning. So just really quickly, I do have a few conflicts of interest, but none are entities that are part of this talk at all. So I often use this slide. This is January 2nd. I leave it blank because it could have been any year for that matter, but this was January 2nd, 2020. And this was a Hong Kong veterinarian, I believe, who posted this. And this is what I saw on Twitter. And this was really the first alarm that I heard of something was coming. And really changed the tenor of the entire year, because I've been planning around this and I've been watching it on a daily basis. In fact, when the first case was presented in the United States in Seattle, Washington, was the day that I was presenting this to our infection control committee and talking about this new pathogen that I was concerned about. Literally, I got a text from my father in the middle of it that said that COVID is here. So I think, many of us would not have known in the beginning of the phases how much of an impact that this would have on all of us. But I think it's important to realize and to look back as we think about the experience, not just now, but in the future, how this will change our practice. So this is where we are. We have 125 million cases worldwide. We've had 30 million cases in the US. The number of those that we've lost and who've been tremendously affected by the disease is an understatement to think how tragic this has been, despite our best efforts to better understand and prevent the disease. But I'm not going to spend time going through all of the history of this, because you guys know this well. I don't think there's really value in a meeting like this with a half an hour to talk. It's impossible to go through everything. So I think I'll spend my time talking about vaccines. I'll talk about variants, and then we can talk a little bit about future directions in where this is going to go and what I think the field might be going towards. So I think, in our cancer patients, we know that with recovering immunity post-transplant or post-treatment with chronic illness, they have poor response to standard dose vaccines. And so I think it's important that there's also heterogeneity with our patients with underlying treatment, their diseases, et cetera. And that many of these novel treatments or novel vaccines are not really studied in high-risk populations, because the expectation is they won't respond well. And if you're a vaccine study, you don't want to have people who don't have good responses. So, timing a vaccine still remains a debate in the field. If you look at flu vaccine post-transplant, 3 months versus 6 months, 3 months in Europe, 6 months in the United States, although that seems to be changing somewhat, we still are not entirely clear. And then we also know that robust seroprotection can be lost in patients, particularly those with allotransplant or CAR T-cell. So, how we follow patients through with novel therapies and the effects they have on people who are getting cancer therapy are not well understood. So we still have a lot to learn about how we deal with vaccines, and imagine a novel vaccine that has a novel mechanism of action. This can be quite understandably a challenge for us to think through. And I think one way to think about the challenges that exist in this space and what frustrates many of us in immunosuppression who deal with immunosuppressed patients is that an example like Shingrix, which was approved in 2017 for the general public, but clinical trials in cancer patients were completed and finally published in 2019, despite the fact that there's evidence that patients with zoster have 2 to 7 times the incidence in the normal patient population. So unlike COVID-19, where we've tried to focus on high-risk populations, particularly those in the BIPOC communities, those who are affected by COVID-19, many times these particular studies are not necessarily focused always on the highest risk patient population, which includes many of the patients that we deal with every day. So I think that's the challenge we're going to continue to struggle with in vaccines. And we currently are in this state where we have approved vaccines through the EUA mechanism, but we have very little data on how to deal with them in cancer patients. So, a typical vaccine, we have an extended period of time where we can begin these discussions and trying to organize, how we approach vaccines in our patient population. And we had a lot of advantages. I mean, we had adenovirus technology that had been studied both in HIV vaccines and had been approved for an Ebola vaccine in Africa. We had preclinical experience with mRNA vaccines for influenza, Zika, and other viruses. None had been approved, but it was actually work in the space. We had experience with protein subunit vaccines with hepatitis B and meningococcus. And we had a lot of these technologies that were readily and adaptable for this. And we had experience with SARS virus, the prior SARS virus. We had rapid sequencing and isolation of the SARS-CoV-2, and we were able to determine the vaccine target much quicker than we would in a normal enterprise. So the science and technology was really there. So we're in a much different situation. And if you would ask me in the early phases of this, whether we'd have a vaccine early, I would have said it's going to be a year and a half at best. And here we are with vaccines at less than a year in time, which is frankly amazing, but is also a reflection of how much research went into these vaccines over time. And we, in some ways, were very lucky that this existed. So we had an expedited, basic science was already pre-existing. We were able to get these things very quickly. And obviously, there was a lot of people who knew the results of these studies because they started doing manufacturing prior to approval process. And basically, these were able to get out to people almost immediately after the EUAs were approved by the FDA, and decisions about allocation were made through the CDC's ACIP committee. So now, of course, these are the sort of spectrum of vaccines, and I've highlighted the ones that are EUA approved in the United States at the moment. There are the vector vaccines, which include AstraZeneca, the J&J, and the Sputnik vaccine, which is the Russian vaccine. There is mRNA vaccines. We have the protein subunits vaccines and the killed virus vaccines, which include Covaxin, which is produced in India, and the Coronavac, or the one made by Sinovac, which is a Chinese version. And these are all slightly different. As an example, the Covaxin is an adjuvant as well as a killed SARS-CoV, where Coronavac is just the killed SARS-CoV. I'm not entirely sure. We don't know the details of when Novavax or AstraZeneca will be presented to the FDA, but we at least have 3 that have been approved through the EUA mechanism. So I'm going to talk about the ones that at least are somewhere in the pipeline. There's a varying range of efficacy against COVID-19. So 76% with AstraZeneca. This was recently revised from their public statement, which said 79%. And then J&J is somewhere between 60 and 70%. Moderna 94% and Pfizer 94%. And again, this is against COVID-19 infection or disease. But hospitalization you see and death, these are actually where people will describe 100% protection. I think it's really, you have to be very cautious about this particular statement. I think that feels really good to talk about that, but when you look at the actual data, there are so few that actually die or are admitted to the hospital in these studies that I think as time goes on, this will not be true. We will continue to see some people be hospitalized and some death in these vaccines who've received these vaccines. I just assume that the numbers and the percentages will go down substantially. Now, remember, each of these has a slightly different cold chain. Pfizer's gotten a little easier now, it's -20. And then the dosing strategies are similar in most, except for Johnson & Johnson, where they do a one-time dose. In the trials, there is the Ensemble trial was the Janssen J&J initial study. They are doing a vaccine study where they're doing 2 doses of the vaccine, which I think may end up being better. But we don't have that data yet, and that probably will not be finished until this summer. And I will say most of these, except for the Pfizer, go down to 18 years of age, but there are additional studies in children, which I think are going to be critical for protecting pediatric patients with cancer, who really at this point have no barriers beyond what are masking, et cetera. So, vaccine guidance has been a little bit funny because what we normally base our recommendations are our clinical trial data or prospective non-randomized data. We make recommendations based on other groups with immune suppression who've been vaccinated or on similar vaccines that have been used. We sometimes make recommendations on other vaccines, and sometimes it's just expertise from the community. And as you can imagine, it's been the latter. It's been recommendations that we've based these data on from vaccines from other areas and from expertise from the community. So we've used prior examples for things like influenza, but we know that they are less robust in cancer and transplant patients, and that high dose vaccines or boosting more than what is recommended for the standard population may be better. And then we also know that responses can be present in some of those that are the most highly immunosuppressed, even 3 months post-transplant. So a lot of this has been where we've sort of built these data to make recommendations on a national level. So, NCCN guidelines right now are for 3 months post-auto and allo, or for cellular therapy patients and after cellular recovery post-cytotoxic chemotherapy. And then the recommendation for all others is to give vaccines whenever eligible or available. But this doesn't feel very comforting, because I think many of you would understand that the patients who get Rituxan or patients that are getting other novel regimens, we don't really know how effective these vaccines will be. But I think the key in this committee was to focus on access. We really felt like urgency of vaccination and avoiding barriers to getting vaccinations in our patients was critical. To really advocate for early vaccination and cancer and transplant patients. And I early on volunteered to sort of help lead this effort, partially because I was concerned that nationally we were not seeing a movement towards cancer patients being at the top of the list. And we wanted to get these guidelines out to be able to use this as a tool for advocacy for our patient populations. And while it hasn't worked out perfectly, I've seen many states across the United States have moved quickly to put some of these patients at the top of the list. I would love to say that the aspects of how we have delivered vaccines across the country has been smooth and organized, and that cancer patients have been early on that list, but that has not been uniformly true. In fact, I think many of you would agree that many of our patients are still clamoring to get vaccine, and we're not quite to the place where we have enough where everyone can get it. So there still remains challenges in how we deliver vaccine, and I hope we can learn from what we did wrong and what we did well with this process in the future. But clearly there is a major gap in data and understanding of how these vaccines work in our particular patient populations. So what we do know is that there were cancer patients enrolled in these clinical trials. Pfizer had about 1,395. Johnson & Johnson had 226 cancer patients, and interestingly enough, had 79 patients who were immunocompromised after a blood transplant. But data on these subgroups is not clear. They've not been made available. I have reached out to both companies to get additional information, and as of yet have not been given that information. So we do not know at this point where and what this data suggests. And I think my guesstimate is they are working on this, but we don't have that data yet. But I think it's interesting that there were some of these patients who did get through the system. Now I'm not sure if these are all patients who are post therapy and are long-term survivors, and that's probably my expectation. But even that data would still be helpful for us to be looking at. 79 patients who were post-transplant seems like a massive number in the short term, and would have been very helpful in planning how we move forward. So what do we know about early data and responses? And there's one of the reasons I put my slides together very early. I've learned through the SARS-CoV epidemic that I try to do them as quickly as I can, sometimes even the night before I give a talk, because the data changes so rapidly, as you all know. And this data recently was published in medRxiv. It's not been peer reviewed, but at least provides some early data. This is a UK study from Oxford and King's College, and They found that, that about 59% of solid tumor patients, but only about 13% of hematologic malignancies responded to the first dose with detectable antibodies. And you can see in that image there, you see the spike-specific immunoglobulin that it's in the hematologic malignancies on, I guess would be your far right, is significantly reduced compared to the healthy donor, healthy controls. Solid tumors were also slightly lower, but not quite to the same level. But interestingly, solid tumors provided good boosting, where they had about 90% with seroconversion and evidence of virus neutralization. They didn't have sufficient numbers in the hematologic malignancies to comment, but they did see some boost in some of those patients. And then most interesting, which I think is really critical, is they did see some interferon gamma and IL-2 producing T cells that were specific for SARS-CoV-2 antigens that appeared to be somewhat attenuated in cancer patients, but also appeared to be improved by that boost. So I think there may be some data that suggests that there is potentially some benefit. The question is, how much was that boost, and really how high the levels are the antibodies? What is the level of protection, and what is the role of T cells in controlling the disease? So lots of questions around this, but I think it does provide some evidence that these vaccines do have some efficacy. I think we'll find out more as additional groups add to this data. I think one other group that you can look at is other immunosuppressed populations. And I think Brian Boyarsky at Hopkins, for those of you who are on this call from Hopkins, did a really impressive study of solid organ transplant recipients. And the way that he did this sort of mirrored a little bit how you guys have organized yours. It was very much a process of sharing research links with the general community, where they have over 2,000 solid organ transplant recipients who volunteered to be a part of the study by sending in blood samples from all over the country. They had 436 in their first portion of the study, and only about 17% had detectable antibodies at day 20 post-vaccine. So that's about a fifth of a normal host. Second dose data has been submitted, but is not yet available, so that will be interesting to see if they have a similar boost. And there's no available T cell data, because that is, as many of you know who process T cells, it's very difficult to assess T cell data from a distance because they have to be processed so quickly. So I think they'll have some data from their local patients, but very much less from the general population. And most were greater than 2 years post-transplant. So the population is less immune suppressed than most active cancer patients. So it's going to be hard to sort of know how these data will be relevant for our patients. But I think it's another immune suppressed patient population that gives you some sense that the vaccine responses are going to be somewhat attenuated. There are no data that I know of to date that have been available for the Johnson & Johnson vaccine, but I think many of us would argue that a prime boost strategy where 2 doses are going to be probably better for this particular group. So what about efficacy against variants? Well, there's data that continues to be developed in this space. The mRNA vaccines did not include data on variants, and I think it's really important to understand that when the mRNA studies were done, the data on variants was just not available. So these finished before many of the major variants were reported. But they did show live virus neutralization against the B117 variant from the UK, the B1351 variant from South Africa, although less in that particular viral variant. And then the adenovirus vaccines with Johnson & Johnson did include Brazil, which was a P2 lineage, and South Africa, the B1351 variant. And those, again, showed they were less effective, but they still did prevent severe disease and death, which is obviously critical. AstraZeneca, as a comparison, did not appear to protect against particularly the South African strain when compared in a pseudovirus or neutralization assay, but did appear to be effective against the most common B117 variant, although a 9-fold decrease in prevention and neutralization. So variants of concern are sort of interesting, and they continue to sort of develop. The nomenclature isn't really easy to understand, so I put the names in there just so you have that reference. But this is sort of where we are at the moment. We have the B117 from the UK, So a 50% increase in transmission, increased hospitalization mortality, and reduction in post-vaccine neutralization. You have the B1351 from South Africa with, again, increased transmission, known higher viral loads, and actually a prolonged period of shedding of the virus in a normal host, with, again, reduction in post-vaccine neutralization. Same for Brazil with the P1 variant. And the new California strains, which are P1427 and 429, again, an increase in transmission, although not as dramatic, and again, reduction in post-vaccine neutralization. I will point out that in Seattle, we're starting to see about 25% of the cases that we have are California variants, and we are seeing increases in cases in the last few weeks, which is a little bit concerning as we've seen this around the country. The other variants of interest are out there are the B1526 and B1527 from New York, and the P2 Brazilian variant, but those have not met the variants of concern criteria from the CDC. And there are, at this point in time, no variants of high consequence, and those would be ones that either are not effective, vaccines are not effective against these strains, or they have a change that would indicate that they would not be picked up on a classical PCR. So that would be a change in the focus area where the PCR would anneal. And I think those are going to be important ones that we'll be on the lookout for. But as of the moment, there are none in this particular situation. So as you've seen the strain development over time, and if you haven't gone to Next Strain, which is developed by a colleague of mine, Trevor Bedford, I would really encourage you to play with these data. They're incredibly interesting, and do go to the CDC link to make sure you understand which ones are which, because they have different nomenclatures for phylogeny for these viruses. But this has been the sort of strategy of how they've looked at different viral strains over time with influenza, West Nile, and now COVID-19, that you can see how this diversity has really shifted and how now variants are really the most important, and how some seem to have more dominance than others. You can see the B117 from the UK is really taking over a large portion of episodes in the world, where the Wuhan 1 strain is really faded in comparison. And then if you look at frequency and variance in US cases, you can see that the B117 variant just over the last month has increased fourfold. So I think there's a lot of concern from many of us that this will continue to expand, and one of the reasons we've been pushing for a vaccine to get out so quickly. So I think variants are going to be something that we need to be very cautious of. And I think when I thought about this, this paper that was recently put out on medRxiv is one that I think is probably the most important in terms of how we think about this. So you think about post-vaccine antibody levels, there's the protective efficacy and percentage. Following a vaccine, there's a natural decay in antibody titers. And if you look at the figure here, these are the sort of estimates of protective efficacy, with Novavax and the mRNA vaccines being on the higher end. You see the Sputnik vaccine, which is that R-Ad26 and Ad5. You have convalescent plasma, you have Covaxin, you have the Ad26 Johnson & Johnson, the Chadox with AstraZeneca, and the Coronavax, which is Sinovax, or the Chinese vaccine. You can see the spectrum of how that efficacy is, but that efficacy will drop over time. And if you really model this, I think the way to look at this is if you look at time when a booster would be required. So when they have, when you have a 90% efficacious vaccine, 70% efficacy, it will drop to 70% efficacy at about 131 days, so about 4 months. And then at 221 days, about 50% efficacy. But if you have a vaccine that is 70% efficacious, you'll drop to 50% efficacious at 90 days. And so these are pretty remarkable differences, and may suggest that there may be needs for boosters in patient populations. And again, efficacy here is really looking at the development of COVID-19, not necessarily death and hospitalization. But this will remain important as we think about how we can prevent transmission within our communities. So I think one concern for cancer patients is this really the perfect storm, where antibody levels at the beginning will be less robust. That decay will start from a place that's not quite as high. So that lower peak will lead to less time when antibody levels would be considered protective. Variants already have reductions in post-vaccine neutralization, And so that combination of low vaccine levels or low antibody levels, followed by variants that are more apt to escape and need higher vaccine levels, may really put patients at high levels and may lead to less robust response to vaccines in our patient population. And then if you look at things like IVIG, not really sufficient to provide protection at this point, but something that we'll be watching, I think, closely. So we don't have a way we can easily add something to the mix, at least one of our usual therapies to provide protection. So the other thing is that immunosuppressed patients continually are being discussed as a source of variants. They are the ones that do have shedding data for long periods of time. They have higher viral loads. They have virus detectable, and then with that limited immunity, they are more ease of viral escape. So if you have components of the immune system that are knocked out, it allows the virus to escape what we would normally expect. So I think shedding a viable virus may increase that possibility of transmission as well, but that's something we don't know. So a couple of papers as examples, and I refer you to McCarthy's science paper from the University of Pittsburgh, and then one that Jesse Bloom did a few years ago in eLife, looking at influenza. So I think, when is this all over? What should we change? What did we learn from all this? And I love this cartoon from the UK. When this is all over, what should we change? And the answer to that is everything. So I think there's lots of questions about vaccines in the future. We need immunologic data. I think there's no question in my mind that boosters will be needed. The question is how often? And I think, will we have enough vaccine to make that possible? I think eventually we will, but something we're really going to need. Is one vaccine better than the other for particular populations? Is it a combination of two vaccines? Should it be an mRNA vaccine followed by an adenovirus vaccine? Do we need higher or different doses of vaccine to improve responses? And do T cells provide sufficient protection? And are those with limited antibody protected? I think these are really critical questions we're going to need to understand. I'm sure that many of you on this call have already seen patients with cancer who have progressed through and have developed disease. So I think we need to continue to discuss who these patients are, what they're developing, and how they're breaking through, and what their antibody levels are. I think we really need to focus on caregivers and family. We know that household transmission is high. Caregivers and family can't leave their homes. They have high risk exposures. They can't prevent that exposure. We need to focus, I think, differently than we've done in the past, is to really spend time doing cocoon strategies, where we vaccinate those who have known efficacy in family members, and not those who have not focus as much on those who have unknown efficacy, who are cancer patients. And I think we need to see renewed efforts from cancer centers to target household contacts and caregivers for vaccine education. We've had, a study that was done from Emory suggested that few cancer center websites even target any vaccine information or education to close contacts. So that is something we have to shift on for sure as we move forward. So I think the expectation is vaccinated cancer patients will continue to develop COVID-19 when they become hospitalized, despite vaccination. So I think we can see this. It'll probably be less, but I think we need to be wary of that. I think decreased community levels will decrease risk, but regression to more vaccine coverage over the year and over the next 2 years is going to continue. We'll see more vaccine hesitancy develop. I think similar to H1N1, COVID-19 will become a continuing issue that we're going to continue to see. And I expect a yearly vaccine for all of us, and maybe even more frequently for our patients. I think the variants will continue to see breakthrough coverage. Will they break through particular vaccines? Don't know. Will they lead to missed laboratory detection that will lead to subsequent outbreaks? Could be critical to know. We don't know. We haven't seen that yet. And how will variants play into future vaccine boosters? The Moderna vaccine is already testing a new vaccine that will include a South African strain that targets that specific spike protein. So I think there's incredible science that's able to move that much faster. And where will the next major variants of importance be identified? And I really think, if you think about it, this is where the worldwide distribution of vaccine is. And there are plenty of places in the world where there's not adequate vaccine, particularly Africa. And I think novel strains will be developing in these populations and will cause mischief and mayhem for sure. I think we need to learn more about treatment options. There's a lot coming. I didn't spend a lot of time on this, but monoclonals, the new Merck drug that's EID 2801 or molnupiravir, fluvoxamine, which is an antidepressant agent, which has actually shown some benefit, could be a preventative. And then monoclonals for prophylaxis for high-risk patients, I think is going to be quite interesting. There are some new novel antivirals that are in the pipeline, and then eventually IVIG and how that could be useful. And I just posted this information about the molnupiravir study that was presented at CROI a few weeks ago. We're at day 5, looking at data in patients who were infected, although in small numbers. There was clearance by day 5 of virus, so this is quite exciting. I also like to think about how we can go back and do things differently, and I would just say we've done such great work. We've protected our patients, we've done education, we've shared protocols, we've developed national guidelines, studies have been developed on a shoestring, vaccine delivery systems have been improved, and we've really focused on equity amongst our system and vaccine allocations to high-risk populations. We've shifted how we think about masks. I think we're going to be doing them for a long time and every year for quite an extended period, and I think vaccines will continue to learn a lot about what we do. I think we can learn how to prevent other viral infections. This is data from Seattle where you see major reductions in other respiratory viruses with all we've done. How can we parlay this into continued efforts to prevent influenza and other complications? And I think we need to learn from pandemic pathogen preparedness. We need to share our data. We need to continue to improve our communication. We need to avoid silos, that we don't do our data by ourselves. We fight presentism so we can avoid our staff from coming in and transmitting and leading to outbreaks. I think we've learned a lot about telehealth, and I encourage all of you to focus on advocacy for patients and diversity in our research. So I think the next wave, I hope, and I think what we're talking about today is really the next wave of science and the next wave of research that's really going to help our patients be better treated for COVID. And I am hoping that what we learn from COVID will help us to protect patients in the future beyond just COVID, but for other infections and other complications. And at that, I'll stop. It's a lot to get through in 30 minutes, but I hope I got enough across to most of you to help get this started. Thank you so much for the invitation.
Thank you so much, Steve, for a wonderful presentation, a really amount of data, and thank you so much for your contributions in the field and impact on patient care. I will try to go through a few questions quickly. We don't have too much time. I cannot probably go through all of them, but let me ask you, Dr. Enstoff, my colleague from the Cleveland Clinic back in the day. Mark, I hope you're doing well. He's asking about in the data from the Monin-Aldana publication, what was the breakdown of lymphoid versus myeloid malignancies, if you remember that part?
Yeah, it's actually pretty small numbers from that study. I think it's about 14 patients total that were in hematologic group, and I think if I remember correctly, it's about half that were myeloid and half that were in the other that were lymphoid, but I can't remember off the top of my head. I'd encourage you to go check it out. It's a really interesting paper that was just posted a couple of days ago, and I think will really help us think about it. Much of this is modeling, so I'm not sure how it will reflect reality, but you know, we have no other way to sort of look at that. But I think it's the I'm talking about the vaccine data. Sorry, that this was actual true data, but there's not enough data in this that we really can estimate. I think there's just too small a numbers in that study for hematologic malignancies. But I think that data is going to be coming like a firestorm as centers continue to enroll these patients.
Thank you so much, Steve. Maybe we'll tackle one more, and then I will ask you if you can tackle some of the other questions in the live chat. Sure. Yeah. Dr. Advani is asking actually a three-way question, three parts. Mm-hmm. Have some data of the current vaccination rates for patients with cancer in the US, and if there's any specific groups declining vaccinations. Mm-hmm. Also, he's asking about any, you showed some data, we don't have much, about immune responses in patients with cancer.
Mm-hmm. Yeah, so I think I'll tackle the first one. The immune responses, we just don't know yet. I think there's lots of people collecting data on this. Many of you on this call probably have small pilot studies where you're collecting information. I think there's going to be larger studies that the companies are going to do in these populations. There's, as an example, the CIBMTR, which deals with transplant patients, is planning a large study to look at collecting data on these patients. So there's going to be some natural history studies where patients are going to be vaccinated. They're going to be antibodies looked at. They're going to look at outcomes. I think that data is going to be coming over the next few months as this gets out to patients. I think what's hard is to understand a little bit about declinations so far, because there's been so much diversity in how the vaccine has been allocated to cancer patients. So in the state of Washington, as an example, we've been doing people 65 and older, and other places around the country are making this available for everyone. So it's really hard to comment on this. I would say that my sort of general sense is that cancer patients are so excited to get vaccinated that they're bothering their physicians every day to figure this out. But there's going to be no question there's going to be some hesitancy. We're just not sure where they are, and I think it's going to be diverse, like all other hesitancy has been around vaccines. It's going to be across multiple different populations. Certainly in Washington, we have high levels of vaccine hesitancy in certain areas. But I also think this is a novel situation in the pandemic, and I think people who have often been hesitant have been more willing to get vaccine. I think the safety data also is quite substantial in these vaccines. They look really good, and so I think patients are becoming less and less concerned.
Thank you so much, Steve, for an amazing presentation. Again, work overall in this field in the last year and all your help. There is a couple of more questions in the chat room. If you have a,
I'll take them on. You got it. I'll try to go to the Q&A and answer a few of them if I can.
Fantastic, Jeremy. This is back to you. Thanks again, Steve, for your amazing presentation.
Thank you both. That was great. So